Good morning. Today's session is about the photosynthetic gas exchange, that is the gases that are exchanged during photosynthesis in green plants. We have seen in earlier sessions that, the plant takes water and minerals which are found in the ground, and of course, it takes them through its roots. It also takes the carbon dioxide from the air. Moreover, sunlight is absorbed by the chlorophyll, which is a green pigment, found within the chloroplasts of the plant cells. And then, the plant cell uses these elements in order to synthesize glucose, which is an organic substance. The glucose molecules will be transformed into other organic substances, but most of the glucose will be stored in the form of starch. In addition to glucose, photosynthesis results in the production of oxygen gas, which is released into the atmosphere. We will start by watching a video showing that plants release oxygen. At the end of the video, try to answer the following questions. 1. How can we prove that the collected gas is oxygen? 2. Is this experiment alone enough to prove that oxygen is produced due to the occurrence of photosynthesis?
This activity is to show the production of oxygen during photosynthesis in the presence of sunlight. The materials required for this activity are beaker with pond water, hydrilla plant, stemmed funnel, test tube. Consider the apparatus shown in the image. Take a beaker and fill it with pond water. Place water plant like hydrilla or elodie inside the beaker and cover it by a short stemmed funnel. Take a test tube and invert it over the stem of the funnel. Ensure that the level of water in the beaker must be above the level of the end of inverted funnel. Place the entire assembly of apparatus in the sunlight for at least 3 days. It is observed that in place of water, air is filled in it. This air is actually a gas and is collected in the test tube. Test the gas collected in the test tube by inserting a glowing incense stick, which would burst into flames. This indicates the presence of oxygen. The air bubbles formed in the test tube is of oxygen gas. Thus, it is clear from this activity that oxygen is produced during the process of photosynthesis in the presence of sunlight.
Now, let us answer the questions. How can we prove that the collected gas is oxygen? A wooden stick is lit. We allow it to burn for a few seconds, then, we put out its flame. While the tip is still glowing hot, the stick is introduced to the gas sample. If the gas is oxygen, then, the wooden stick reignites to produce a sustained flame. Now for the second question. Is this experiment alone enough to prove that oxygen is produced due to the occurrence of photosynthesis? Actually, this experiment shows that the plant can produce oxygen, but we cannot be sure that this oxygen gas produced by photosynthesis. In order to verify that oxygen is the product of photosynthesis, we have to stop photosynthesis, that is, we place the plant in conditions that inhibit photosynthesis. In this case, if oxygen is no longer produced, then, we can be sure that the oxygen gas is produced by photosynthesis. This experiment is conducted to indicate the gases exchanged by a green plant in light and in darkness. The variation in the volume of oxygen is measured using an instrument known as an oximeter. This instrument monitors and records any change in the amount of oxygen. The results are shown in the figure below. You have to know how to read the oximeter results. When you see the volume of oxygen decreasing, this means that oxygen is consumed, but when you see an increase in the volume of oxygen, this means that oxygen is released. Now let us read the results. During the first 10 minutes, the plant is placed in darkness. During this time, the oxygen volume decreased. Of course, this means that oxygen is being consumed. But then, we place the plant in light, from the 10th minute to the 20th minute. And upon exposing the plant to light, the volume of oxygen increased, so, oxygen is released. But after that, the volume of oxygen started decreasing upon replacing the plant, back in darkness. Now, before answering the questions according to the Lebanese curriculum, let us think about the variable factor. In this experiment, we have placed the plants in different conditions, which are light and darkness. So, we can say that the variable factor in this experiment is the presence and absence of light. Of course, when we analyze, we have to look at the 3 different phases of the curve. We should mention the variable factor in each phase, and describe the variation observed between the beginning and the end point of each phase. In this case, we have to say, the volume of oxygen decreased below zero during the first 10 minutes in darkness, however, it increased to reach 0.3 millimoles, when the plant was placed in light. After that, we mention the third phase, which is the decreasing phase, when the plant was placed once again in darkness. Of course, we should use a comparative connector between the different phases to show that we have difference in results, so we can use the terms, however, while, etc. Then, we have to give a conclusion, which should be an answer to the posed problem, based on what the results have proven. In this experiment, the question asked was, what are the gases exchanged by a plant, in light, as well as, in darkness? So, according to what we have seen, we can say that, in light, the plant is releasing oxygen, while in darkness, the plant is consuming oxygen. Therefore, our conclusion will be that plants release oxygen in light, but consume oxygen in darkness. Please pay attention to the answer. The key words are written in bold, and are underlined. During the first 10 minutes, when the plant is placed in darkness, the oxygen volume decreased below zero. However, upon exposing the plant to light from the 10th minute to the 20th minute, the volume of oxygen increased till reaching 0.3 millimole, while upon replacing the plant in darkness at the 20th minute, the volume started decreasing, reaching 0.2 millimole during 5 minutes. Of course, the conclusion is, the plant consumes oxygen in darkness, while it releases oxygen in light. Graph analysis includes conditions, including the variable factor, values, which are the numbers representing the beginning point and the end point of each phase of the curve, units, description of the variation, that is increasing phase, decreasing, constant, fluctuating, etc. Comparative connectors. Moreover, if the graph includes more than 2 curves, then, our analysis should show correlation between these curves. We will see this in later sessions. Look at this figure, and try to distinguish between photosynthesis and respiration. Please note that photosynthesis is represented in white, and respiration in red. Pause the video and try to answer. So as we have seen, during respiration, glucose is used to produce energy, while photosynthesis uses the energy from the sunlight to synthesize glucose. During respiration, oxygen gas is taken by the plant, and carbon dioxide and water vapor are released, while, during photosynthesis, carbon dioxide and water are consumed, and oxygen gas is released. Moreover, respiration process takes place at all times, that is during the day and during the night, while photosynthesis occurs during daytime only. So based on what we have learned, the overall chemical equation for photosynthesis would be carbon dioxide plus water, in the presence of light gives glucose and oxygen. The overall chemical equation for respiration would be glucose plus oxygen gives carbon dioxide and water vapor, and this process liberates energy to be used by the cells. During the day, both photosynthesis and respiration are taking place. We know that photosynthesis is releasing oxygen, while respiration is consuming oxygen. So, how can we explain the increase of oxygen during the day? Most probably, The rate of photosynthesis that produces oxygen is higher than the rate of respiration that consumes oxygen.